This is a brief tutorial on what the genetic algorithm for rule set prediction, or GARP, is as a modeling platform and what kind of a question we're trying to ask or answer with the GARP modeling system. So our goal with ecological niche modeling, and this could be applied to multiple modeling platforms, not just GARP, but in this example we'll talk about the genetic algorithm for rule set prediction. And the idea behind this modeling approach is to link environmental conditions, such as those identified here in 1, what we call coverages in GARP. Notice here I have a set of covariates that describe temperature, precipitation, elevation, annual vegetation index, and temporal Fourier processed annual amplitude. These top 2 variables come from the WorldClim data set, and these are ground station interpolated data. Elevation and measures of NDVI can be downloaded from various satellite-based data sets. And then I have here in 2 my species occurrence data. Now GARP is what we call a presence-only model, which simply means that pseudo-absence data are derived from random draws in the background inside the modeling approach, and as a user you only have to provide presence data. In this case we're using anthrax outbreak locations from the continental United States, and the 2 different colors here, yellow and green, indicate a random draw that I've done as the user, subdividing out data that will be used in the modeling process and data that will be used post hoc after the model to evaluate how well it predicted geographically. So those are our input data. The next step in the modeling process is to model the relationships in variable space between the environmental conditions, or some combination of conditions, and those presence points. And in this case, we're doing that using the if-then logic statements known as rules that GARP creates, and we'll look at those again in a moment. Once this model has been constructed, or in the case of GARP, a set of models in an experiment has been created, those rules are then projected back onto the landscape and converted into a presence-absence model that tells us where we're most likely to find the species, or the areas of highest confidence that the species could be supported at that location. If we're confident, after evaluating our accuracy metrics, that the prediction on our native landscape, here, the continental US, is adequate and accurate, we can then replace the environmental conditions from our native landscape with a novel landscape. In this case, the landscape of Mexico, with the same covariates here in 5 that I showed you here earlier in 1. Those covariates can be replaced, or the rule set can be applied to those, and we can project that distribution onto our novel landscape. This novel landscape could be either a novel geography, like here, the same time period or contemporary covariates, and 2 novel landscapes. You could also replace this novel geography with a novel time period. So for example, you could estimate these conditions or some set of conditions in the current and future landscapes under future climate change scenarios, and you could project how the landscape or the distribution of the species might change on the landscape in the future. So let's look briefly at what the genetic algorithm for rule set production or prediction is. In the original 1999 paper, the term production was used, and in later papers, some of the papers most commonly cited today, production has been replaced with prediction. So you'll see it interchangeably, but originally was rule set production. The original model put out by Stockwell and Peters, 1999. So it's a computer modeling approach to pattern match environmental parameters with species point locations, and it does this through constructing if-then statements, which we call logic rules. And there are 4 types of rules that can be produced, range rules, negated range rules, logit rules, and atomic rules. And I'll show you each of these in detail in a moment. It's an iterative presence-absence modeling approach. So this means that GARP is going to work through a process of randomly selecting presence and pseudo-absence points, and it's going to try and match using one of those 4 rules or rule types in a series of combinations called a superset or a set of rules that can be any of those or a combination of those 4 rule types. It's going to iteratively resample and replace presence and absence in the background and try and find the best combination of rules that predict presence and absence. And it's a genetic algorithm because it has the capacity to change, mutate, or evolve rules from previous rules. So GARP is not just building a single logistic regression function or a single range rule, but rather it's building a set of rules, what we call the rule set. And for every individual GARP model in an experiment, every model must have 50 rules. And these rules are created through mutation and evolution. So different parts of rules can be recombined. Individual values within rules can be changed, such as a point mutation. GARP from rule to rule can completely throw one out and start over. Or it can swap the if and the then statement on different rules to arrive at a set of 50 rules that predict the landscape. And as part of that iterative process, it's calculating accuracy metrics at each step within each model to test how well those rules are performing. Because of this genetic nature, this is what we call a stochastic or a random process. And the random process means that we want to run multiple models. So the reality of running a GARP model or a GARP experiment is that you're going to create a set of models and you're going to evaluate across that set for how many of those models agree at any given pixel that that pixel should be present for the species or absent for the species. And so it's what we call a multi-modeling approach. And within that multi-modeling approach, we're creating multiple models, each with multiple rules, 50 rules, made up of a combination of these 4 rule types generated through a stochastic process. So a single GARP experiment contains X number of models, usually 100 or 200, and each of those models has 50 rules. Now what you'll find as you look across papers and over experiments that there's a lot of homogeneity in those rules when GARP is finding good relationships between point occurrences and environmental covariates. So if you have an adequate sample size and you have the right or the appropriate covariates for modeling, you'll find that even though there's the opportunity for a lot of diversity in rules, GARP is relatively conservative in the rules that define the landscape. So let's look briefly at these 4 rule types. We have atomic rules. These are going to be rules that come from the old bioclim models that say if a specific value of an environmental covariate or specific values of specific covariates are met, then the species could be present or absent. Range rules are a bit more appropriate for modeling population-level species distributions. Range rules say if a range of values for a covariate or ranges of values for a set of covariates are met, then the species is present or absent, depending on if it's a presence or absence rule. Negated range rules are the opposite. If specific conditions, or if the point is outside of specific conditions, present or absent. What you'll find as a rule of thumb is that negated range rules tend to be absence rules, and atomic rules tend to be relatively few within a rule set and predict relatively little space on the landscape. Range rules and logistic regression, or logit rules, as we call them, tend to be the dominant rule types across models. And if you look at the literature by Joyner and others, and the master's thesis by Timothy Joyner at University of Florida in 2010, and you look at Alison Barrow and others' work from Sear Lab, where we modeled the distribution of anthrax in Australia, we discussed these rule types and the prevalence of rules in great length. And the logistic regression rules are the application of logistic function where we solve for p, where we have a distribution of 0 or 1, and the covariates are fit with coefficient. Where this is different, of course, is the way that this is populated is by randomly grabbing covariates and trying to fit them. So it's a little bit different than a classic GLM that you see for other species distribution models. But each logistic and range rules are widely used rules across GARP experiments. So here's an example of an atomic rule. Here's the if and the then statement. So it's a logic string. If the following conditions are met. Here we have 3 covariates, bio12, soil condition, so soil moisture, soil pH, and if these specific conditions are met at a given pixel, then that pixel is defined as present. Here's a range rule. Here you can see we have more variables. We have bio 1, 12, elevation and soil, and mean NDVI. And if this range of conditions and this range of conditions and so on is met across this rule, if a pixel meets those conditions and that rule is assigned to that pixel, then it will be assigned presence. Remember, range rules are one of the dominant rule types across models. Here's a negated range rule, and notice the logic statement changes to an if-not statement. So if it's not those ranges met by this set of variables, in this case we have an absence rule. Please note that as we've gone back and forth from the last slide to this slide, we've seen rules predict each presence and absence. And that's part of that draw of constructing that rule, is to select present or absent and see if that rule meets those conditions. Okay. Also, remember, as a rule of thumb, but not always the case, negated range rules are often absence rules. Here's our logit, or logistic regression rule. Here's our if and then statement. So this is a presence rule, where each of the variables in green are included in the model. And we have a coefficient assigned to each covariate. And we have the sign of the relationship. It's either a positive or a negative relationship. Here I've identified one of each to fit this function. So if this condition described by this logistic equation is met on the landscape, that pixel would be assigned a value of present. When we make a map of a single experiment output, we often employ the best subset procedure described by Anderson et al. 2003, and there are other videos on the Sear Lab YouTube channel that describe the best subset procedure. But what we typically find is a single map like this illustrating that best subset, or those 10 best models out of the experiment summated between the value of 0, here in gray, to 10, the darkest value, giving us this gradation that you can interpret as confidence. The darker the color, the higher the agreement between models, or the more confidence that that pixel could support the species. That number 10 is important here because here is 1, 2, 3, and so on through 10. These are the 10 individual models in the best subset that's added up in this larger map. What you're looking at here in the red and orange rules are presence rules identified by a P in the legend, and the blue rules are the absence rules, those areas where the species is not likely to be found. And what you can see across this set of individual models here where the rules are plotted is that relatively few rules, 5 or 6 out of the 50, describe the majority of the landscape. The other thing you can see is that those rules, even though you don't know what's contained in the individual rule here, other than if it's present or absent, but what you can see for this landscape is a relatively few rules describe this northern corridor, and relatively few rules describe these large areas of absence. If we look at the individual rules, which we've done in various studies that can be read, such as Blackburn and others 2007, Joyner 2010, you can explore how those rules are evaluated. But what's important for this particular lecture is that relatively few rules describe the majority of the landscape when there's a good fit between the covariates and the point data. Now, that best subset procedure and the accuracy metrics to evaluate GARP models is described in other videos as part of this series. Those accuracy metrics come from the classic Fielding and Bell literature on false negatives and false positives. In the GARP literature, we refer to false negatives as omission, or we're omitting real data from our spatial models, our GARP predictions. And we call that a hard error because real data points are being excluded by the models. When we do that, we underpredict. Commission is what we call a soft error. And the reason for that is that commission is the percent of the landscape predicted present by a given model or by a best subset. We call that a false positive in that we've predicted that pixel and we don't know if it should be present or absent. Now remember, that's the whole purpose of running an ecological niche modeling experiment. So we have to evaluate commission as a balance between over-predicting, predicting too much of the landscape and not being sensitive enough to differentiate presence and absence accurately, and under-prediction, not predicting enough of the landscape. So let's look quickly at this example. And the way this study is designed, the yellow points were used to build the models. The green points were never introduced to the GARP experiments, and they were used after the fact to measure omission, commission, and the area under the curve, the 3 most common accuracy metrics for GARP models. And let's evaluate what these look like. Notice here I have an area highlighted in northeastern Oklahoma, and that's an outbreak that occurred in the 1950s and had long been hypothesized to have been an industrial outbreak or an outbreak associated with contaminated bone meal. So when I built these original models, we left those points in the model. We didn't exclude them from the modeling process because we wanted to see if GARP would be sensitive enough to model the areas where the pathogen is being sustained, frequently in outbreaks, and exclude that area that was only reported once. So let's look at a common metrics table from a GARP model. Here I have the number of points, the yellow points on the last map, to build the models at 130. And you can see here that those were divided in a 50-50 training testing internal to the GARP modeling system. And if you see the other videos where we teach you how to populate a GARP experiment or how to calculate a best subsets procedure, by dividing this inside of GARP with training and testing data, this allows us to calculate the intrinsic omission for our best subset procedure. So we subdivided that 65 points for training and testing. GARP is allowed to separate those internally. Then we had 47 points additional to use outside of the GARP experiment to test for accuracy. When we did that, you can see here that our total emission from our best subset was about 7%. So we had about a 93% accuracy in predicting our green dots from the previous map. You can see that our average omission was a little bit high, about 23%. And that said that across those best subsets, we had some high omission. And here's our predicted distribution. And the way I would describe this in a paper is to say that there was broadly a north-south corridor from Texas up to the Dakotas, and then a corridor here through the north going to the Pacific Northwest, and again here in sort of the corridor along the border from Texas out to California, with relatively little or no prediction in the central or southeastern states east of central Oklahoma. And notice also that that area in Oklahoma was not predicted by my best subset. So that area of relatively high omission here was good because those points that I included in the model were excluded from the best subset, meaning that GARP was sensitive enough to not identify that ecological conditions in Oklahoma just because there were points there. So we have additional training videos for you to learn about the best subset procedure, but the best subset procedure is the way we arrive at those 10 models out of the 100 or 200 models that we originally constructed within the experiment. And the papers by Wiley and others, 2003, Kluse and McNeesett, each in 2005, introduced a methodology for applying the area under the curve in a receiver operating characteristic or a ROC analysis, and that's now a common metric for evaluating GARP models. So internally, using those training testing split data, GARP is generating a confusion matrix, classic way of taking actual data and predicted distributions and determining each measures of commission and omission, areas of overprediction and underprediction, respectively. And so this happens iteratively within the GARP modeling process to decide which rules it's going to keep and then to decide whether or not the individual rule set is accurate for predicting presence-absence. When we want to do accuracy metrics outside of GARP, when we want to know if that spatial prediction, in our case for anthrax, was accurate, we then subdivide the data before they go into GARP. So I showed you the green and yellow dots earlier. Those green dots, that independent testing data, which is usually between 20 and 25% of your total sample size, are used to calculate a confusion matrix outside of GARP using the GIS, the raster prediction of the best subset and the individual 10 models in the best subset to calculate our area under the curve, and then to calculate our measures of total and average omission and commission that you saw in the table just a moment ago. So we do a post hoc evaluation to calculate the area under the curve.